And everything. All right. Thank you guys for being here and taking your time and energy to travel to Little Rock and to be able to do this. I'm going to try to go through a lot of information. Some of I can just tell from the conversations just on the way over. Some people are new to proteomics, not sure what the resource can do, what we have done, and so we're going to hit this from all different angles here. So Rick kind of alluded to this earlier, and that is that we have a grant, an R24 grant that we get from NIH. Alan Tackett and Mike Hinter are the co-directors of that, co-PIs of that grant for this last cycle that we're just finishing up. And as he mentioned, we're waiting to hear back about the next 5-year submission. But our aims are not just to do basically quality service. Essentially, NIH decided that rather than buying instruments, a lot of people that want to do proteomics, you say, well, you've got to have an instrument. If you're going to do protein mass spectrometry, you need a mass spectrometer. The problem is there's a lot of, you need the skills to be able to run that mass spectrometer. They're very expensive. And then sample prep, there's a lot of expertise involved in that. And so this R24 grant essentially puts money into one place that would have the skills and the already to be able to enhance that for other groups that don't have that infrastructure there. And so first we did across the state, then we did a kind of a regional thing with Oklahoma, and that's where we were partnering with Mike Kinter, who's over at OMRF. And also we provide outreach and education opportunities, which includes the workshops. We talk about a voucher program, which is basically a pilot level free proteomics. And also we have internships that we'll talk about for graduate students, postdocs, training opportunities for faculty members, and all that kind of stuff. So we'll cover all of that. We'll cover all of that. Here we go. So besides those folks that you saw there, there's a whole team of people. Some of you guys got to meet Lisa down there. She's part of our sample prep. We've got a couple of bioinformaticians in Alan and Charity. You guys have been coordinating with Heather. Sonnet's also behind the scenes. We've got a whole bunch of people. It takes a lot of teamwork to be able to pull something off like this. You can't just have, I mean, Rick's a great scientist and does great mass spectrometry, but he can't pull all this up. But you know there's going to be a but he can't do all of this, as you know, and Sam is very meticulous on getting all those details on the front end, but he can't also run the mass spectrometers and do all the bioinformatics and all that. So it takes a big team to be able to do all of these different parts here. So we're located, you guys came in this building, we're up on that 9th floor there, and that's just part of us. So there's also a part, the targeting part, and you're going to hear from Mike Kinter tomorrow about what they do in Oklahoma. But we do discovery proteomics here, which is our job is to identify and quantify as many proteins as we can. Mike and his group is out there at OMRF, and they do targeting, which is basically kind of a validation. They do absolute quantification, and they can do more like dozens. You want to know all of the proteins in this particular pathway, and he would pick peptides for those and be able to do that in a different way than what we do here. And so he'll talk about that tomorrow, what the targeting is. But essentially, targeting proteomics is useful for validating, let's say, 20 proteins or less in a biological sample. That's my short version of targeting. He'll spend a lot more time talking about that. So let's talk about this, what I call the global proteomic workflow here. So your principal investigators are sitting here. You've got some sort of science or biology or disease model that you're looking at, and you want to do some proteomics. Well, you might be asking the question, well, how do I get that? Well, what we do is all this stuff in the blue box is what we do. We do all, let me say, sample prep. There's really, as you're going to find out, there's basically 2 types of sample prep. There's where you guys do the experiment sample prep, right? You house the animals, you grow up the cells, all that kind of stuff. There's some sample prep there. And then there's the sample prep where we take those samples, the frozen tissue, cell pellets, the serum, whatever, and we transform that into proteins and peptides so that we can collect data, do bioinformatics, and we do all of that kind of stuff. And give that back to you. But the thing is that for some of you, you may be thinking, well, so really you're just looking for, you're taking in your sample and you're just trying to identify as many proteins as possible. Well, like 15 years ago, that's what proteomics did. Now we can take and we can do comparisons between groups. So not only are we, the quantitative part is where we identify and quantify, and then we can tell the differences, the relative differences between proteins. And that's really what people are looking for today. And what we spend most of our time doing is what proteins are elevated between the treatment and the control group, the knockout versus the wild type, which ones are significantly lower, which are significantly higher. So some of the experiments are actually done at your lab, and then you just send samples to us where we do our own sample prep. I just want to make sure that's up front, that you don't just say, oh, you guys are going to do all of our experiments for us. That's not the case. So how do we help researchers? So just a couple of things. We do clinical studies. We had a guy that left here a year ago to go into industry, Aaron Story, and he works in a kidney repository group. And so being able to do clinical samples and be able to use proteomics to get meaningful data for the clinicians, that's really exciting. Just in this last year, Stephanie Byram, who recently was our bioinformatics lead, she moved to go work at St. Jude's. She and her team developed this ProteoDA package that we'll talk a little bit, that we still use here, and we'll talk a little bit about that. But being able to develop some sort of downstream package that allows us to be able to give you more information about your data in a rich environment, and we'll talk about that some. And then also the thing is, we have some people that we do work with that goes over spans of years, lots of different experiments. Letters of support come out as they write their grants. Basically, they can depend upon us to do proteomics for them. Professional advice, we do Zoom calls all week long. Sam will take a lot of those. He'll pull in Rick or myself or some other team members as needed to be able to give people the advice they need about how we could help them with their experiments. So I'm going to go through this quickly, and if you've already submitted samples to the core, you've seen something like this, but here you are somewhere out there in the country, and you contact us, usually by a Zoom call or an email to Sam. And I just want to emphasize this again, before the experiment actually has been done. Don't have the samples sitting in the freezer when you call us. That's a bad idea because we can't really help you at that point. Some of those mistakes can't be undone. So Sam will talk about experimental goals and designs and sample groups and comparisons. All that kind of stuff needs to be done on the front end here. Raw samples are sent with a sample list. We'll talk about how important that sample list is, being able to identify what the groups are. And then all the stuff in blue here, this is the stuff that we can do here. When we say sample prep, we mean we get those samples, we do cell lysis. Some people like to do their own cell lysis and send us that, but that's okay. We can also do cell lysis here. We can also do a tissue homogenization. And then we're going to do a protein quant. We do reduction and alkylation. And we'll talk a little bit about what all these things are. All of these different parts, but all what we call sample prep is taking your biological samples and preparing them into peptides. Once they've been digested into peptides, then what we call proteomics is basically, I think, more of data collection, where we want to use those mass spectrometers to be able to identify those separate those peptides, identify them, quantify them. We have spectra of fragments of peptides. And then we take all that information in bioinformatics, then we'll do a search on those peptides, do the protein identification, and then put the whole experiment back together again in this differential analysis that we're going to talk about tomorrow into this final report. and then we send that back to you. And that whole thing, the sample prep and all that kind of stuff, usually takes about 6 to 8 weeks. So when we say proteomics, like Rick had talked about, there's lots of proteomics. You may be in a lab and you say, well, I do Western blots all the time, and that's proteomics. You're right. We're talking about protein mass spectrometry, and we are talking about bottom-up or shotgun, where we are going to take the proteins and we're going to digest them using a enzymatic a reaction with trypsin. And that then those peptides are searched against a database. And that's really important for you to know. If your protein is not in that database that we're searching against, we're not going to find it. And I know that sounds like profound on some levels, but you'd be surprised how many people go, well, actually, I have this list of these weird clones that I expected you to find. Well, if that's not in our database, then we're not going to actually find those. So sometimes people will send us custom databases and that. So let's just get in a little bit more graphically. Again, we're going to just keep covering the stuff in deeper and deeper levels, more detail. You've got biological information in proteins, right? We take those proteins, they get reduced and alkylated. We add trypsin, and those become peptides. Those peptides undergo separation on liquid chromatography. Then we detect them by mass spectrometry to get essentially two pieces of information, two types of information that we'll talk about. One is the MS, which is a scan that tells us things like what the mass is of these ions, what the charges are, and then we can determine which ones are peptides. And then from those, the mass spectrometer is smart enough to be able to single out those and be able to take isolated ions and hit it with energy to fragment it into this MS2 spectra. That information then goes into a database search where we get the peptides, and then those peptides, the ones that are unique, can be matched up in that library against the proteins that we find here. So up in this top guy here, each one of those signals could represent what we call a precursor or a peptide. And down here, each one of these signals represents a fragment of a peptide. And you may be struggling as to what those fragments look like. And again, we're going to get to all that. But the whole idea is that we start off with biological information. We all think about cells and everything as there's information in the DNA. That information gets translated into proteins. And then what we want to do is take that protein biological information and get it into digital biological information that we can then look at and make decisions. Our sample prep basically starts with cells or tissues or fluids. We get homogenization, we get lysate or whatever. The whole goal is to get a clarified lysate so that we can do a protein quant. There's some other parts that sample prep does, but that part right there, that workflow is done over and over and over again. Reduction and alkylation, just to review here. So proteins, they fold up, and then those cysteines will bond together, and these cysteine double bonds. We want to break those with reduction. And then to keep them from reforming again, we do this alkylation experiment. And the important part about that is that the mass spectrometer measures mass over charge. And so we need to know what mass that we're adding. So when we do this alkylation, we're actually adding a mass of 57.02 to each cysteine. So that's something that we can add to our database search at the end if we do that. Okay, so now that we've reduced and alkylated and we want to do a tryptic digestion. So trypsin is an enzyme that the reason why there's other enzymes that you can use, but the reason why we use trypsin is because it's very specific and it's very efficient. And I would like to say it's very cheap, but it's not. If you calculated what the price was, like we buy it in the microgram quantity, I mean, it's more expensive than gold, I think. But trypsin is a protein itself that when added to your sample and you get to pH right, it will go through and it will selectively cleave at the C-terminal of the lysines and the arginines. So remember, there's 20 amino acids, 20 normal amino acids, and two of the basic amino acids, lysine and arginine, that's where the trypsin will go in and cleave that. So this picture I have over here, I have made it color coordinated so that the blue and the red will show you where all of the lysine and the arginines are that the trypsin would cleave. So out of this particular protein that I have right here, this is human insulin-like growth factor 2. It's a 605 amino acid in this protein, and you'll end up with 70 possible peptides. So trypsin's really good because it's lysines and arginines occur about 10% of the time in proteins. So lysine is a good choice. Trypsin's a good choice because it will come and cleave this this protein, which is really big and unwieldy and sticky, into smaller pieces that we call peptides that we can then make solutions out of and do analysis with. Because one of the big questions people always say is, why not just analyze the proteins themselves? Seems like we're just making it a lot more complicated by breaking proteins down into peptides. Well, again, proteins are sticky and they're hard to analyze in mass. Peptides you can make solutions out of, and solutions are much easier to deal with. So it only takes about 2 unique peptides to end up with a score above the predefined threshold to identify a protein. So some of you might be like, so do I need to analyze and find a signal for every peptide to say that protein's there? And the answer is no. We just need a couple of unique ones that only go to that protein. Some of these guys are going to be too small. They're going to be too small for the mass spectrometer to pick up the way our chromatography is set up and all of that, and we will never see them. Some of them will also be too big, and they won't, we would say they don't fly well. Mass spectrometry is about getting these peptides into the gas phase with a certain charge on them so that we can route them through all the different places in the mass spectrometer. So I want to talk a little bit about chromatography here, because that's an important part, and it's a part that basically frustrates a lot of people that try to do protein mass spectrometry, is that there's some chromatography on the front end, and we do nano LC. So that means we do, we'll have flows at the nano level, 300 nanoliters per minute or so. But there are thousands of peptides generated from each tryptic digestion, okay? The goal of the nano LC is to separate those peptides into groups or bands so that we can analyze all of the peptides of the same type all at the same time. Because if they're all spread out, then you're going to get, it's just going to look like a bunch of noise. We want to, that's what chromatography does, is it gets them into peaks. So the separation is done by hydrophobicity, which means the more hydrophilic, those are usually the smaller, more highly charged peptides will elute first, and the hydrophobic ones are larger and they're less charged, will elute later. You crowd those peptides into concentrated groups that can be analyzed at the same time. You get a bunch of them concentrated together, and now you get a much bigger signal that rises above the noise, and you can then quantify that. To get those peptides to have a charge, we resuspend them in acid. Usually it's 0.1% formic acid, or is it 0.01%? It's 0.1. It's 0.1, sorry. So they will be charged. So there's some chemistry there, basically leveraging the chemical properties of peptides to be able to do that. So this is a really ugly picture, but I wanted you to see that, you know, it's just like a race car. You see this pretty race car on the outside with all it's all shiny and everything, but if you open it up, it's the engine inside where all the work is done. It's got hoses and tubes and pulleys and all that. It's the same thing. Nano liquid chromatography, we've got a bunch of valves with a bunch of plumbing here that we have to take care of. And the reason why nano LC is so hard is because at 300 nanoliters per minute, to get one drop, one just normal sized drop, it takes almost 3 hours. That liquid is flowing so slowly. And so the plumbing tube sizes, the dimensions of these tubes are ridiculously small. They're capillary size, and the pressures are enormous. So we have a lot of things going against us. And if you get an air bubble, it's not just a bad day, it's like a bad week, because it takes a long time to get an air bubble out of a system like that. So once we get through sample prep and those peptides we digest the proteins into peptides, and those peptides get resuspended into the right solution. We will put them in a plate like this, and those peptides are now dissolved into this solution, and then we would take that little bit and inject that using an auto sampler. And you can't see this very well, but this red line shows that's the column. That's where all the separation occurs. It looks like a wire. It's not much thicker than a human hair. And it contains, it's really a tube full of this resin that is separating those peptides out by their hydrophobic ability, and then shooting that into the mass spectrometer. And so this is a picture here. This is the very end of the column. It's hard to see, but there's a resin that's inside of this. The tip of this is about 10 micrometer in diameter, and the liquid is spraying out. And then there's a little opening or hole here where this is the mass spectrometer. And so you can think about it, it's vacuum over here in the mass spectrometer. That's why it was so noisy in there. And it is spraying out liquid here. And that liquid, the peptides are being drawn over because of charge. And we'll show you a picture of that in just a minute. So that tip of the column, it's like 3,000 PSI or so. We're flowing at 300 nanoliters per minute. And then the inlet is under vacuum over there. So technically, there's a lot going on, a lot of physical processes going on to be able to get those peptides in the right state, at the right concentration, separated into the right groups to be able to get introduced into the mass spectrometer so the mass spectrometer can do its thing, which is a pretty amazing thing that it can do. Well, how are those peptides separated? This is just a basic picture here. The LC system is made up of these really expensive pumps that can deliver high pressure and low volume and very reproducible flow rates. That's it's $100,000 with a pump, basically. And then those peptides are separated with a column that I showed you that's got a resin with particles down on the micrometer size. So if you were to look at it, it's just like a little sphere here, and hanging off of that sphere, we've got a C18. This is a hydrocarbon tail off of there. And this may not be exactly what it looks like, but it's pretty close. All of the resins are proprietary by the different groups here, but the resin particles are covalently bonded with that C18, and that causes, as the peptides come along, it retains those peptides just a little bit so that they will separate into their groups. I know I'm going through a lot of chemistry and stuff here, but I just want to give you a kind of a taste of different things as we go on. And if you want to go dive into deeper, Rick would love to jump into all of this at some level. So this is what's that? Bore you for years, and I'm just going to do it for a few minutes. So this is a picture here that shows the LC. I don't know if you can see this, but the LC over here, this is where that liquid is coming out into what this is what it looks like. And remember, all those peptides are going to be charged. And so we actually have a power supply with that is providing, I'm having a hard time with this, providing about 2,400 volts here that is drawing those liquids out, and they come out with a droplet. But those peptides are surrounded with solvent, water mostly, and in that small little, about a centimeter distance here, those solvent droplets evaporate out until we just get gas phase ions. This is a peptide with a +3 charge, let's say, going into the mass spectrometer. Okay, so the physics of it is we don't want all that water, we don't want all that solvent. That's just gonna that's gonna complicate things. And so there's a lot of chemistry going on between here and here to be able to get those gas phase ions into the mass spectrometer. Well, the mass spectrometer, what exactly are those components of a mass spectrometer? Well, the black box here, and I stole the slide from Rick, is essentially you've got an ionization source here, and that's where you get basically liquid samples have to get into the gas phase, and we just talked about that. There's some gases that are flowing along that are getting rid of the solvent. Then you have a mass analyzer, which is a vacuum chamber that somehow separates molecules that differ in mass. So these ions will differ in mass, and it's able to separate them out or identify them. And then you have a detector, which takes those ions and is able to convert that into some sort of electrical signal that can be measured. And so the more ions that you have, the larger the electrical signal. And then you've got to have some sort of data system that can basically, by the time, has something to do with the electronics of the mass analyzer, will indicate what the mass to charge of the ion is, how long it's flying through there. There's all kinds of different things that it can calculate to be able to say, this is an ion of a certain intensity, of a certain mass to charge ratio that is hit, and be able to keep all of that data straight. This is what the inside of a Orbitrap Tribrid mass spectrometer looks like. And unfortunately, I don't think I got I'll show you maybe the animation tomorrow. I don't think it's going to work today, so I'm not going to try it. But I'm going to walk you through here, and maybe for you folks I'll do it with a mouse here. Let's see. So the ions will come from here. These are these little balls here, and they will come into the mass spectrometer. That's that little junction point where you have the end of the LC and the beginning of the mass spectrometer. There's a funnel here that essentially focuses down, just like a camera does, where they have optics. They also have optics in a mass spectrometer. It focuses down the ion beam, and then it does a funny thing. It goes around a corner. And the reason why it goes around a corner is because anything that's not charged will not make this corner. All the neutrals will fly this way. All of the ions will fly around this corner. You have a quadrupole mass separator here that will be able to identify and give you a scan. You have those ions then can be selectively put into this next, what's called an ion routing multipole. And those guys can either then go into this area here where it gets hit with more energy, breaks it apart into fragments, and then measures it on the ion trap. Or it takes those ions from the ion routing multipole, takes them back into the C trap and kicks them into the orbit trap, where they then will oscillate around this spindle until it measures the frequency of that oscillation and gets an exact a high resolution mass. I'm not making this stuff up. This guy will do this hundreds of times a second, and dozens of times a second, and it can do things at the same time. So while some ions are being broken up, some peptides are being broken up into fragments and analyzed over here as the MS2 signal, other ions are being routed into the orbit trap and are measuring the high resolution mass of the peptide itself. So there's a lot of stuff that's going on, and this is an amazing, it's an amazing instrument that does that. And they all work slightly differently, but that gives you kind of a picture of what that is. How are we doing on time here? Okay. Well, what does the initial data actually look like? And this is always the part when you go into a mass spectrometer lab and you see one and you see this like weird trace coming off there and you're like, what am I looking at? What am I seeing here? Is it good? And you got to look at a few of these for a while before you kind of get the hang of it. But the idea is this is called a chromatogram here. It has relative abundance over there on the y-axis, and on the x-axis it has time, usually in minutes. And most of our runs, we now have an instrument where we do 20-minute runs, some 40-minute runs. A lot of our instruments will do an hour-long run. And that's the amount of time it takes to separate those peptides out into all its different components, get them into the mass spectrometer, and then analyze those. And so you'll end up with you could have 100,000 peptides in a single sample. And the goal is to have those peptides spread out over that time window by a gradient. And so you might be wondering, well, how do you get those peptides to separate out? The gradient is that the hydrophobicity of the peptides are all different. So we will start with a very aqueous mobile phase, and we will gradually change it to more organic mobile phase. And as the organic character changes, that will cause more of the peptides to come off of the resin and go into the mass spectrometer. So the ions that are down here at this end are the more hydrophilic ones that are coming off first, and the ions that are coming down here, these are the peptides that are going to be more hydrophobic here. And what we want to do is we want to spread these guys out so that we can, but still have them in very sharp, narrow peaks so that we can measure them. So where are the peptides? So if you choose an abundant peak from that chromatogram, like where this little line is here, and you can you can zoom in on this guy and you can tell by the isotopic signal that you get here. Some of you might remember from your chemistry that there's these things called isotopes, right? So carbon is this amazing one because 1% of all carbon is carbon-13 instead of carbon-12. And so since all peptides are made up of carbon, then you're going to end up with, naturally, you're going to end up with a signature that looks like this, where you have the parent signal that has all carbon-12 carbon-12 atoms, and then you have another signal that would have just one, maybe one carbon-13 atom in that molecule and one with two, etc. So each one of these signals, again, there's two pieces of information that we get from the mass spectrometer. One is the scan. You can look at the bottom here, and we're scanning. The M over Z goes from about 400 to 1,000. And that full scan, each one of those signals, there are groups of signals, could represent a peptide. And if we were to look in at an MS2, the MS2 signal, I don't know why this didn't come up here. There we go. Let's just get it all up there. So the MS2 signal, so it basically, most mass spectrometers that we have are for doing proteomics are doing something like this. We'll take a scan, an MS1 scan, and then we'll do a bunch of MS2 scans for a few seconds, maybe like 3, and then we'll do another MS1 scan, and then we'll do a bunch of MS2 scans like that. And this MS2 scan is telling us about the identity of a particular peptide. So this guy back here, each one of these signals here represents a different peptide. This guy here is a particular peptide that has been fragmented, and it fragments, believe it or not, you hit it with just the right energy, and the major fragments are these Y and B ions up here. And the Y and B ions break at very specific spots so that you get a loss of mass that is equal to the amino acid. And so back when Rick started to do protein mass spectrometry, what a protein mass spectrometry guy did would be get these MS2 signals and do some math to figure out what the identity is of the peptide by calculating the difference between these different Y ions. And so the difference might be alanine, the mass of alanine, or valine, or whatever. Computers are really good at that, and they will process these things in bulk and be able to measure this for us. So you get an output now. There's lots of different software packages available, open source and commercial, that will show you what from the spectra that you have from your mass spectrometry run, it will match that to a database. So here's that same protein that we were talking about before, that human insulin-like growth factor. That's the whole sequence. And when I ran my sample and I analyzed it, all of the signals there in gold up there, those are all the peptides that was actually found that passed the threshold that matched this particular protein. And this is one in particular here that you see on the screen, is this peptide here, and you can see all the Y ions here identified in blue and the B ions identified in red. And you can see above the spectra here, you can see it's actually showing you which amino acid it's categorizing to because of the differences between these Y ions and B ions here. And there's a table form over there, and there's some other things with the chemistry. You can have protons that are added, you can have water or ammonia that's added or taken away. So but those guys there confirm, there's enough signal there to confirm that peptide is identified, and that peptide comes from this particular protein. That's a lot, but as a biologist, you need to know this, because someone's going to say, well, you did a proteomic experiment. How do I know that you actually found the protein that you're interested in? We could say, well, we found some signals that match to a certain peptide that are unique for my protein, and that's done all the time. There needs to be some other validation with that, of course, but this is where proteomics is that you're relying on the fragmentation pattern of these signals to be able to then infer the proteins that are in there so that it'll back up the biology that you guys are actually doing or looking for, okay? I stole this from Mike Kinter here. He's really good at doing an overview here, but he said proteomes are known and finite. And I think that may be changing over the years, but in essence, what that means is that you can go to a library and at uniprot.org, and you can look up Homo sapiens, and they give you the instruction code for every protein that would be in a human. There it is, right there. And so if you were to look, you can say, well, there's 20,000 genes, give or take, and it has a protein count of 80,000. So there's a couple of different libraries that you can download, but we take all of our data. We don't do, what do they call that? Ab initio with de novo. We don't do that where we just try to figure out everything from scratch. We take we peptide signals that we get, and we compare that to a library. And so that library will have, in this case, just like we saw before, it has a name up here at the top, which is like it gives you some information about that particular protein, the gene it comes from, a little description, and then it gives you the primary sequence there. And it does that for all the proteins that are in there. What it doesn't tell you is it doesn't tell you where all the sugars are hanging off of and the phosphorylation or other PTMs there, right? It just gives you the primary sequence. So we might get something like this, a list of protein IDs that as an output based upon your different sample, and that same information that came from the UniProt ID will then get split up here to where you have the gene, the protein group and the gene name and the size of it and all that kind of stuff. Okay. I'm going to take a pause for just a second here. And so we're going to kind of like circle back around here to kind of put this all together, because a lot of times we gloss over this. But we can start with a gel. People will do separate proteins out on a gel, and we still do gels here. We can do like one of our workflows is a digestion workflows is a chloroform methanol extraction. So if you give us a cell pellet, we can use that, or we can do a filter aided sample prep. These are all digestion techniques. All of these things essentially are a sample prep to get peptides, added to trypsin to get peptides, all that are charged, that then go through the same LC separation process to go into a mass spectrometer to become gas phase ions to be able to get MS1 and MS2 signals. Now, that's not all the story for all the workflows, but that is, in essence, the workflow that we're talking about. The sample prep is all that stuff up there to get peptides. The proteomics is to get to this, and then the bioinformatics starts from there on. Okay, so a couple other things we're going to talk about. So we said some post-translational modifications can be analyzed. We do phosphorylation here, and it's actually really interesting, because if you'll do the enrichment on your end here, and you tell us what the modification is and on what amino acid, the bioinformatics, the data search part is actually kind of easy. If you say, I just know that every time you come up with this amino acid, there might be this group hanging off of there, and I've enriched for that, then it's really easy for us to go in and put that modification in the database search. It's really hard to do the enrichment, though, and that's why we have you guys do that at your end. There are some groups that do glycosylation. We are not one of those groups. And then there's some other things here. So when people say, well, okay, so is every experiment the same? sometimes I'm not seeing what I want to see, or how do I know your experiment is going to be the right one for what I'm looking for? So each experiment's designed to quantify and identify the greatest number of peptides and proteins in the biological sample. And what we're really doing is we're looking for candidates, right? If we're doing a quantitative experiment, you're essentially saying, I've got this group of samples over here that have a phenotype that's different than this group over here. And I want to figure out what the proteins are that are different between those. There won't be a lot. Maybe there'll just be a handful. Maybe there'll be a few dozen. And so sometimes if you don't see the candidate you're looking for, it's because it's really low level. And we can always increase the depth. That means get more proteins identified by doing fractionation and more instrument time. And that would be a Rick specialty there, where he would look at it and he'd say, you know, you're looking for some transcription factor. There's only 1 or 2 of those molecules per cell. You're only giving us 200,000 cells. We're just below the detection limit. So we either need to use a different instrument. We need to fractionate this, which will help concentrate that, you know, something like that. So that's one of the challenges, is that you just don't see what you want to see. Here, another challenge, if we look at the plasma proteome as a good example, is that if you look at the abundance, the normalized abundance for different protein groups as a function of these different classes of proteins in plasma, the problem is that we have a large range. It's more than 10 orders of magnitude difference between some of these proteins that are really abundant, like albumin, and some of these guys down here that are really interesting that you want to take a look at. And I love this example. Rick will tell people, it's like going out, well, you can't because it's cloudy, but it's going out on a sunny day and trying to count the stars. Well, you only see one star, right? It's the sun. The sun is blocking out everything else. And that's what happens in the mass spectrometer. If you don't have a way to remove albumin or some of those other really abundant things, you're never going to see some of this, the low-level things that you're interested in. And so we have some depletion protocols and things that we can do to help with that. But that's one of the challenges of proteomics, is that you see highly abundant things a lot, and you don't see the things that you're really looking for. Okay, as I wind up here, a couple of things I want to say is when people that are used to doing genomic sort of experiments, they're always a little taken back when we say, we think you need to have an N of 5. And they say, an N of 5? That's a lot. I was thinking of an N of 3. Well, that's okay. You can send us 3 samples per group. And sometimes that works. For cell culture, that usually works. For tissue, that usually doesn't work. For patient populations, forget it, right? So we would say for a good proteomic experiment that you have enough statistical power, try to go with 4 or 5 at the minimum, and then you ask fewer questions. A lot of times people say, well, this is my first proteomic experience, and it's like when you go to the West Coast for the first time and you suddenly want to see the Grand Canyon and you want to go see the Redwood Forest and you want to go see the Golden Gate Bridge. And so suddenly your trip becomes this massive trek and you forget that these things are thousands of miles apart. It's the same thing with proteomics. Stick with a simple experiment that asks a few obvious questions. And that tends to work out better. Then you can get more, you can get fancier as we go on. I want to go through a couple of the highlight the main global proteomic workflows that we have. This is a really busy slide, so I'm going to show this all up here at once, and then I'm going to break it down for you here. But we essentially have 3 different versions or flavors of proteomics that we do here. We have the DDA, which we call the data dependent acquisition. It's up there at the top. That's good for higher sample numbers. The problem is it's a stochastic sampling, which basically means that it favors more highly abundant peptides. It will give you an MS1 scan of all the precursors, and then an MS2 scan. And it's the same workflow. You've got proteins that get digested into peptides, and it gives you those two guys that you're looking for, the list of the range of peptides, the scan of peptides, and then breaks it down. Piggybacking on top of that is our TMT experiment, okay? So TMT is it means tandem mass tag, and that's where you add an isobaric tag It's a very specialized molecule to each one of your samples so that it has a very specific molecular weight tag on there. We also do offline fractionation. We do multiplexing on this. And we will also get MS1 and MS2 information, but with the TMT experiment, we also get MS3. So we can also hit it once with energy to fragment it and hit it with more energy to break off those tags to get quantification. So that the TMT experiment is also one of the advantages for us right now is that is our way of doing phospho proteomics, is we will do a TMT experiment, and then we will do in parallel with that a phospho experiment on those same samples. The experiment that we do a lot of as well is called a DIA experiment, the data independent acquisition. It's really good for serum and plasma. We do a lot of IP experiments or enrichment experiments with that, bio-ID. And the nice thing about it, it's very comparable to the DDA experiment in that there's not a label, but it's equally sensitive to low abundance. And the way it does it is rather than going up here on the top for a DDA experiment, when it makes that scan, it chooses the highest signal peptide and analyzes that one first, then the next one, then the next one, then the next one, until it runs out of time, and then it takes another scan and starts that process again. For a DIA experiment, it's essentially, it doesn't do it that way. It scans the same windows every time as it goes through the range from stepwise from 400 to 1,000. And by doing that, it's insensitive to these highly abundant peptides that can drown out the signal of the low-level stuff in a DDA experiment. The downstream processing and being able to look at the data on the back end is a little bit more complicated with DIA, but it's gotten much better in the last couple of years with different software packages. So here is just this broken down into each slide here. So again, for DDA, the MS signal, each signal there represents a peptide or a precursor, and the MS2, each signal there represents a fragment of a peptide. So the way I think about an MS2 scan is this MS2 scan is like a peptide fingerprint that tells you the identity of the peptide. And the MS1 signal is basically telling you it can tell you some of the quantification information as well. Yeah, I think we've all covered that. Just kind of the workflow that we talked about with the tandem mass tag experiment is we start off by adding these tags here. And I don't know if you appreciate that, but by covalently bonding onto the N terminal of every peptide, a specific tag there, then we can add all of those samples. So you might have 10 samples. Once they're tagged, you can add them all together. And then you can, because you have a way of being able to tell which peptide comes from what sample because of the tag label that's on there. So then we can do this offline peptide fractionation. And all that means is we have another liquid chromatograph that will separate out the peptides into fractions. And that will help enrich and concentrate those fractions before we measure those on our Orbitrap Eclipse. We usually use the Max Quant as our downstream database searching before we go into our bioinformatics there. For the DIA, talk about fractionation here. Yeah, I think, okay, let's move on to cost. I think I'm about done here. So just to give you some idea of cost, I think we've got a new pricing structure. So Sam will correct me if I'm wrong here, but I believe everything at this point, so for a DIA analysis, if you were to have 40 samples and we were to do the digestion here, you're just to send us the samples and we do all of that, I think it's $71 a sample. Is that right? $71 a sample? Yeah, for So for 40 samples, it would cost $2,800. For a TMT analysis, because you have these really expensive labels, sorry, Casey hears this, but those labels from Thermo, they're really expensive. But we do a tryptic glycine digest. We do the labeling. Then we have offline fractionation. And so it comes to be about $196 per sample. So for 10 samples here, it's a little under $2,000 for TMT. And then for phospho, if you add phospho onto that, if we'll end up getting an experiment, the TMT experiment with the lysates, and then we'll do our phospho enrichment as well. And the total cost of that goes up to $3,200. So let's talk about the bioinformatics or the deliverables just on the end here. So we've talked a little bit about sample prep, talked about the workflow, the time, data collection. We've sort of hand waved about the mass spectrometer and how it works a little bit. Bioinformatics deliverables, what do you get on the back end? So first of all, the ProteoDA software that we use will create different things like these. These are all quality control plots, and we're going to go through these tomorrow, that can show and basically help reassure you of how well your experiment went. Our bioinformatics people are really good at being able to say, did you by any chance do this experiment on different days with some of these samples? And then you find out well, actually the graduate student thought it'd be best to run all of the controls on one day and then run all the treatments on a different day because there just wasn't enough time. And the bioinformatics people go, yeah, we can tell. And the problem with that is that you want that biological information to be the only thing that's causing this variation. And so you want to try to minimize those batch effects. So that's some of the stuff that will affect that, that we can see from that. The other bioinformatic deliverables would be these volcano plots, which are really good at being able to show us those candidates that I was talking about, right? In a volcano plot, you have some of these guys, these little lines here represent kind of our predefined areas where we say, okay, if it's up in this quadrant here, that means it's upregulated and it's significant. And those are the things that we're going to look at first. If you're a graduate student, you don't want to get too sad. If you have hundreds and hundreds of things up into these quadrants here, that just means your work is forever. You're never going to graduate, right? So what you'd like is to be able to have an experiment where you've got dozens of things to look at, but not hundreds of things to look at. You also will get a spreadsheet that is got some of the annotation data as well as some of the statistical information that you can go through. And this is and there's some columns here that you can help sort through your information to be able to pull up things easier. That's also some of the deliverables that we have here. So you've got this whole workflow and all of these things that we've just talked about here that we're going to spend over the next day and a half here. And you might be saying, I've got all kinds of questions, right? You just questions everywhere. And so we're going to spend some time over the next day and a half here peppering with our knowledge and expertise at different areas here. And but really the question is where do I start? If you're sitting in the front end of this now, if you're a wizened veteran at proteomics here, maybe this is just a good refresher for you. If not, maybe that what I just said over the last 30 minutes or so is like an avalanche of information that you need time to process. But that's okay. We're going to go through all of that again in a little bit slower. More detailed things. But where do you start? And that's why I like Sam to come here, and he's going to speak to us next. Is that Sam, being our resource manager, we like to call him the front door. He's the guy that gets a lot of questions where people, he's really good at being able to kind of draw out from you what your biology is. He's very good at knowing biochemistry and biology, and has a lot of experience with lots of other projects, and can help you decide experimental design and what workflow to use and all that kind of stuff. And so that's where we that's our front door guy right there. And at this point, I'd like to take any questions if you have any.
So Dennis somehow got a hold of my driver's license photo for this slide. I don't know how that happened exactly.
It's really the best picture we have of Sam. It is. And so I use it constantly, hates it. Yes, ma'am.
Yeah, I have a couple questions. Number one question is, you mentioned that the electrospray ionizing, what does that exactly do?
Rick?
So electrospray is the way that we're forming ions from peptides. And so mass spectrometers measure ions. And we like to say that they measure mass, but they actually measure mass to charge. But electrospray is a process that was invented, discovered, however you want to use the term, by John Finn in the 80s, where you can take a liquid that's got a voltage applied and spray a peptide solution from that, or a protein solution. And as the droplets come out, there is some charge on the droplet. That droplet will, just by undergoing collisions with just the background gas in the atmosphere, those collisions cause the solvent molecules to slowly is a relative term, to boil off in the microsecond time scale. And so you eventually have, a big solvent with maybe a few peptides. And as that solvent comes off, the charge is on the surface of that droplet. And as the surface gets smaller and smaller, as you're losing, molecules to the vapor, the charge becomes so concentrated that you end up getting a Coulombic explosion. So that the positive charges are so close together that you get a little micro droplet that ejects from this bigger water droplet. And that little micro droplet will contain one peptide. And so that micro droplet continues to desolvate until now you're only left with a peptide in the air, and it has charges anywhere that will hold a charge. And so for a peptide that's been cleaved by trypsin, you'll have a lysine or arginine on the C terminus. And so that's a site that will hold a proton. The N terminus will hold a proton. If there's a histidine in there at this pH, because we're working with like pH 3, there'll be a charge on that histidine. And so you may have a peptide and it may have 3 charges. And so that process going from solution, peptides in solution to an ion, a peptide ion in the gas phase, where you haven't put any energy in for it to break apart. It's called a soft ionization technique. And so that was the big discovery for electrospray. It was a way that we could first really look at peptides without having to put lots of energy in. Before that, people were using lasers to hit something with just laser desorption, and the peptides would absorb a certain amount of energy, but then fragment because you couldn't get them to the gas phase. But electrospray allowed us a process to go from peptides in solution to peptides in the gas phase, so a mass spectrometer could measure them. So that's the electrospray process.
I see. That's a very impressive. Yeah. So second question is, my lab is very interesting. I'm studying phosphatase, so phosphorylation is my top priority. So in which form sample should I send you guys to analyze? For example, I have a cells has knocking in overexpress one specific. I'm a study PP2A, so some of those regulatory subunit changing the phosphorylation of some target protein. So in my case, sometimes induction, like a cell quality, has to be reached a level like a perfect induction or reduction. So I needed to validation my experiment work before I can send you the sample for analyze. So
Yeah,
how
so
to do
for something
one?
like that, you'd probably want to validate by, by lysing and doing, the simplest thing is to do a Western blot, right? If you've got-
Yes,
if
yes,
you've got an antibody
phosphorylation.
that shows, in your control state, you've got an intensity at some level, and then in your induced state, you've got a higher level.
Yeah.
I mean, having some biological validation, not just in a phospho experiment, in any experiment,
Yeah.
having a positive control that the experiment worked. That's a much cheaper way to determine whether an experiment worked than to send it to us and we go, you know what, we identified 8,000 proteins and none of them are different.
Yeah.
That means the experiment probably didn't work like you wanted it to, but that's an
Yeah.
expensive way to find that out. Yeah. So, getting any biological readout, but for this, yeah. So you probably got an antibody that you could read that.
So
And
I
then
would
for-
way to give you the samples.
So a frozen cell pellet would be the way to do it, and you'd probably want to include phosphatase inhibitors so that when we thaw that pellet,
You can protect it.
That's right,
Mm.
that's right, and we're going to add them as well, but it prevents any issue. FedEx doesn't always deliver as quickly as you think. And so this would be something that would need to be shipped on dry ice. And I don't know if we're going to talk about this, but
Yeah.
you always need more dry ice than you think. We very rarely have gotten samples with too much dry ice, but almost every week get samples with not enough dry ice, especially in the summer. It sits on the tarmac in Memphis when it's 100 degrees. It doesn't take long to start boiling through the dry ice.
How much is cell pellet? How much cells you normally you have?
So for a normal proteomic experiment, we like to process 100 micrograms of protein. For phosphorylation, we're going to start with at least twice that much.
So
And so, at least 200 micrograms. And, send us a little more if you can, because we're going to do a protein quant. We're going to normalize to that, but we're going to normalize to the lowest sample that you send us.
So
So if you send us
500.
6 samples and 5 of them have 200 micrograms and one has 130 micrograms, we're going to normalize to that 130 micrograms, but the data would be better if we could do it on 200 micrograms.
yeah, so half a milligram should be sufficient.
Yeah.
Right?
So, but if you're going to send cells, you want about 5 million cells.
5 million. Okay, that's good.
Depending on cell type, but yeah,
Yeah.
that's a good number for mammalian. That's a great number to
Okay,
start with. Yeah.
that's okay. Good.
Bacteria, sometimes you might need more. Yeah.
So another
Okay,
question.
I'm,
Okay.
let's